and so we left at this this idea of K-selected and R-selected species. And I was saying that the K-selected species tend to be long-lived, big species, big animals specifically. This is basically designed for animals. Plants have different strategies. But for animals, this is true. Species that have long lives, that have a small number of descendants, they tend to have more stable dynamics, population dynamics. So their populations don't change much through time. So when you look at the graph, you will see relatively stable numbers looking like what you would get from the logistic curve. So these would be K-selected species because we assume that they have strong intraspecific competition that makes their numbers stable. Whereas R-selected species don't really care about the capacity of the ecosystem, and so they produce many more descendants than those that are necessary, right? So if the population is to be stable and you assume sexual reproduction, then you only need to have each couple to produce 2 descendants during their lifetime. And of course, if a population of a frog or a rabbit is going to be stable, then that's what's going to happen. And so this means that these R-selected species have huge mortalities. Most of the descendants produced are dying. But again, if there's an opportunity, if there's a new resource, if there's a new space, then they can grow very rapidly. Okay, so that's why they are selected. They put their money on being ready to take advantage of any new opportunity, and they grow very quickly. Of course, when the resources are exhausted, then they die off very quickly, and so their dynamics is much less stable than those with K-selected species. Now, marine mammals are all of them K-selected species, right? So they're big animals. They produce a small number of descendants, not every year. And so one of the consequences is that they are very sensitive to changes in resources or changes in their lifestyle parameters, including death. This graph, and we are going to talk about this in the next class, but I wanted to show this graph to talk about the current situation of marine mammals and specifically of cetaceans. This is a graph. You can see the references in the presentation that shows an estimate of the number of whales killed by industrial whaling during the last century. Okay, and you can see that in the peak of the whaling, after the 30s, after the decades between after 1920, there were huge numbers of whales that were killed by the industrial whaling. The whole lightening of entire cities was made possible by burning whale oil, and so the slaughter of whales was incredible. In the peak, you can see there for the northern hemisphere and for the southern hemisphere, the number of whales killed per decade, and in the southern hemisphere, most of the slaughter was based on sperm whales, including the Azores, where some of you are, and in the peak of the hunt, you had more than 250,000 whales killed per decade, which is about 25,000 whales killed per year, most of them sperm whales. In the southern hemisphere, that was even worse. In the peak of the season of the years, like you see that, more than 400,000 whales were killed per decade, more than 40,000 whales killed per year. The overall more than nearly 3 million cetaceans were killed, in the largest hunt ever in the human history. And this made the populations collapse completely. So what you see there towards the end of the century was not due to moratoriums or the stop of the fact that the whaling was stopped. The fact was that there were no more whales to hunt. And so when we get to the end of the 1980s, or the beginning of the 1980s, you have the formation of the International Whaling Commission, and you had the moratorium, but that was because there were no more whales. And so now presently we are in a situation, and so let me see, 40 years have passed since the moratorium. The moratorium was established in 1982 and to start into effect in 1985, if I remember correctly. And so but my point here is that cetaceans in particular are in very bad position because they're case-selected species. Their populations grow very slowly. So although almost 50 years have passed since almost 40 years have passed since this moratorium, since the commercial whaling stopped, many populations are still at the brink of extinction, like blue whales in the Antarctic populations of blue whales. They have been reduced to less than 90% of their original numbers. So population then, so this is one element that is important to have in mind in terms of population dynamics. Now, the rest of the lecture, and we really don't have much time, but let's see what we can do, is about methods, right? About how do we do this? I mean, and the first thing is, how do we count marine mammals, right? Of course, this is a basic ecological question. Any ecologist needs to have tools for counting living creatures, and in small populations you can count them all. I mean, if depending on how you define your population, you can try to count all the individuals in a population. But once you have a realistic population, you cannot expect to count all. So what you do is you subsample. You take a sample and you count it in, and you try to extrapolate from that to the whole population, right? And for instance, in pinnipeds, and particularly in otarids, where they have populations that breed, and so you have periods of the year where all the animals congregate on certain areas. You can go to those areas and count the animals that are there, and then you so you have an estimate of how large the area is, and you go to certain beaches and you count them, and then estimate how big the population is, which you didn't count. So pinnipeds have, at least otarids have, this element of at this critical period where it's relatively easier to count them. But if you're talking about cetaceans, for instance, or even many seals, that's not so easy, right? They don't congregate, so they're out there in the ocean. It's very difficult to find them. So what you use is techniques based on transects, and because their population areas are so huge, you normally use either boats or planes even. So this slide here shows flight paths for transect for counting. I don't remember what species of whale is. So, but how do they do it? I mean, they fly over a certain area and they look for whales and mark. So imagine you are flying, or you're on a boat doing these transects, and you're supposed to count whales. right? So the boat is moving, the plane is moving, and you're looking out and you see, oh, there's a whale, oh, there's two whales, and you count them. At the end of the day, at the end of the program, do you think can you say that you've counted all the whales?
I think it's impossible, but
why is that impossible? I mean, you're traveling and you're seeing the whales and you're counting them. What's difficult there?
They spend most of the time diving, so it's just casuality, casualidad that you see them.
Yes, that's one big point. That's one important point, of course. And the other point is that you do not cross. You when you do a transect, you have space between. You can see there. You have spaces where the plane doesn't fly, where the boat doesn't go. So there will be whales there, right? So you have these problems. You only count the animals that you see on a certain range, okay? And of course, when you do that, all the animals that are below the water, you don't count them. So you have to have an estimate of the number. So it's complicated to have an estimate of the numbers. And people have tried to do this, and they have developed methods to deal with this. One of them, and most of the counting of marine mammals in the sea is done using this distance sampling technique. In a distance sampling technique, you don't simply count the animals that you see, you also record the distance that they were from the line you're traveling, and you also calculate the angle that you see them. So say you're going north, and you see an animal to your right. So you have to calculate you have to estimate how far the animal was, and also the angle that this observation does with the path you're traveling, right? And the reason for this is that at the end, you want to have a distribution of the number of animals. So you want to calculate the distance from the animal to your transect line, so you can have a distribution like this one here. And the reason, so what does this mean? This means that you will see all the animals that are on the transect because they're just in front of your nose. But as the animals are farther and farther away from the transect, farther and farther away from your nose, the probability of not seeing them increases, right? So at the end of the transect, you have a graph like this. If you plot the number of animals you see as a function of the distance they were from your transect, you will see something like this. And they're very interesting, decreasing, right? And they're very interesting mathematical techniques in these distance sampling methods, where you use, let me see if I can explain this. You use the animals that you see at the distance to estimate the number of animals you didn't see close by, right? So basically, you look for what they call an effective strip width. So, and you use the numbers of animals that are beyond that effective strip to estimate the numbers that were inside the strip, but you didn't see. So it's an interesting mathematical technique that is widely used and makes use of all the information we have. So we do not assume that we see all the animals. We do not see all the animals, but we use the animals that we see less to estimate the animals we didn't see. Okay? Of course, and to this you have to have another estimation, which is how many of the animals you couldn't see because they were underwater, right? So say, for instance, in sperm whales, you would have, if you estimate that they spend, say, 30 minutes below water, under the water, and they stay 10 minutes at the surface, then you can you will only see the animals when they are in these 10 minutes at the surface, right? And so you can use this balance between 10 minutes and 30 minutes to estimate, again, the number of animals you didn't see because they were diving. So that's the best thing we can do in distance sampling. This is very widely used for sampling animals in the ocean, calculating their densities. The other method that is also very used is mark recapture. You may have heard. Mark, tu já tiveste alguma... Mark, did you have any... have you ever studied these mark recapture techniques?
The previous one, no, but the recapture, yes.
Mark
I
recapture.
did it in ecology.
Okay, can you explain briefly how this works?
You take some animals from their habitat, you mark them, and then you release them again in their habitat, and you come back and recapture the same species, and you note how many of those are marked by you previously.
Yes. Yes, and then you use the percentage between those that are marked and those that are not marked to have an estimation of how many of the animals are in the total of the population, right? So if you captured 10 animals, if you marked 10 animals and then you release them, and they're supposed to mix with the others. So say you go there and you capture 10 animals, you mark them and you release them again, and then you go on the second time and you collect again 10 animals and one is marked, right? Because you have marked 10, this means that for each 9 animals, for each one animal that you marked, there are 9 others that were not marked, that were in the population. So this means that your population is probably around 100 animals. Okay, so the thing is, we cannot capture whales and tag them. So we'll see, well, let me move on. So what you do with this is, the way you do this is by photo identification, right? So photo identification data sets have been used as a sort of mark recapture. Well, not as a sort, as a development of mark recapture methods, where people use the resightings of the same animal to estimate the total population. These are sophisticated methods, not as simple as the one we've just shown, because you have to take into the consideration of migrations and eventually death of the animals. But that's what you have, and so ecologists make do whatever tools they have. Now, so that's one thing. For abundance estimations, these are the two main tools used. Now, the other thing in terms of population dynamics that is important to consider is movement, right? Because animals do not stay, well, some animals are mobile, and particularly marine mammals are highly mobile, and cetaceans are extremely mobile, and many seals also. And so you need to know where are they going if you want to understand their ecology, and if you want to think about their conservation, you need to know where they're going and why are they going. And of course, photo identification is one technique where you can do this. So you take pictures of animals, you build your catalog of animals you saw in a certain region, and then you compare your catalog with the catalogs from other people elsewhere. And you can see, oh, this whale was here this year, but then it was in the Bahamas, and then we saw it in Scotland, and then you start having an idea of the range that the species occupies. And so that's one piece of information. Of course, you can have more sophisticated pieces of information. And now we have tags, satellite tags, that you can attach to animals, and they can then be used to follow the animals for a day, for months, and you have an idea of their movements, and you can collect all sorts of information. So more and more this is expensive, but it's a very useful technique. And if you look carefully to this image, you will see that the concern with the welfare of the animals is involved here, because these tags have suction cups, so they do not attach to the flesh, to the blubber of the animals. They just stay put by suction. Now, the other thing, so that's basically how we look at movements for photo identification and tagging. Another element which is important for population dynamics is social structure. We've discussed social structure the other day, so in addition to photo identification, for instance, where you can follow the animals and see how they interact with each other, there are now more sophisticated techniques using DNA, right? And so one not so nice way of so you need to collect DNA from the animals, and one not so nice way of doing this is just shooting them with a small dart, with a small arrow that removes a little bit of their skin and their blubber. I have seen these darts. They have a stopper, so they have something that stops the dart, the arrow, from going too deep into the animal. So they just collect this little piece of blubber and they float at the surface, and then people collect it, right? And from this you can get very important information for population dynamics. And you can use lots of markers, but for this presentation, the two most important are these. You can look at mitochondrial DNA, right? And mitochondrial DNA lets you look at subpopulations. So because this is a section of the DNA that changes very slowly, relatively slowly, and so it can give you an idea of subpopulations, and it can also give you an idea of the demographic history of the animals. For instance, that's how we know that just by looking at the mitochondrial DNA, that's how we know, if we didn't have the record of the whaling history, when we look at the mitochondrial DNA of present whale populations, we see that the variability is very low. So this means that in their past, there has been what geneticists call a bottleneck. So a period where the population was very small, was reduced, and then it started growing from there, but it started growing from a reduced stock. So the mitochondrial DNA can be used for these kinds of assessments. On the nuclear DNA, you have another tool which are called, for some reason, microsatellites. And what microsatellites are repeated and presumably useless pieces of DNA, where you have long pieces of DNA which are mere repetitions. They're not supposed to encode anything. They're just there, as far as we know. But then these sequences change very rapidly, supposedly because there's no selective pressure on this. So mutations just accumulate, and there's little mechanisms to correct those mutations. And these microsatellites change so fast that they can be used to trace parenthood, so to know who is related to who. So it's the the same technique that is used in paternity tests for humans, and it can be used, and it has been used for marine mammals. Okay, so it's almost 9:30 there, so we have some more time, I think. So, what do I have here? Okay, so now, the other elements of population dynamics that we need are summarized in this graph. This is not, it's a lousy image, but basically we've covered some of that, right? So we've covered movement patterns, we've covered the number of individuals, many areas contribute to population ecology. So for instance, knowing about feeding ecology, knowing about mating strategies are important elements that allow us to model the population dynamics of the species. Now, and I'm going to cover just briefly a few other of these elements that we see here on the presentation. The first one is age. In order to build a model for a population, age of the animals, of each individual animal, is an important goal, right? So assessing the age is important now, and it's also extremely difficult. For most methods, you need to kill the animal to know its age, right? So so normally age determination is done after the animal is dead, and this can be done using basically three techniques. The first one is using teeth, and the reason for this, I could have started there. I have a slide. I have a slide of some later after these next slides, where we can show that marine mammals, and particularly cetaceans, they do not stop growth. So that's an important element. You cannot use, so you can use teeth from odontocets, obviously, to evaluate how old they are. And the reason for this is because teeth keep growing, right? In our case, for instance, we grow until we're 18 or something, and then we stop growing. And our teeth are born with their final size. So there are no growth marks in our teeth, right? Like there probably are not growth marks on the teeth of lions or dogs or cats. I haven't looked at this, but I don't think so, because they grow until a certain time and then they stop growing. Marine mammals, and particularly odontocetes, continue growing. They grow almost like a fish. The growth is, we can assume that the growth is continuous, but asymptotic, which means that they grow continuously, but they grow slower and slower as they get old. But anyway, this means that their teeth are also growing. And so you can cut the teeth and look at the marks in the teeth. And this has been established scientifically using markers, etc., to know that it is possible to calculate the age of a dolphin by counting growth marks in their teeth, right? For baleen whales, one technique that has been used is using the wax in their ears as an estimation of age. The middle ear is blocked, is closed to the outside, but there's still a production of wax inside the ear, and this accumulates with age. So there have been attempts to use this accumulation of wax inside the ear as an estimation of age. Again, for females, I don't have a slide here. Also for females, the number of pregnancies recorded in their ovaries is also used as an estimation of age. And this gives us indications that of how long animals can live, right? And so many whales can live several decades, 40 years, 50 years, 60 years, as far as we know. And one particular whale, which is the Greenland right whale, or Balena mysticetus, can apparently live up to 200 years. And how do we know this? We know this using a technique, a very special technique that is used to date paleontological findings, remains of animals that can be dated using this technique called amino acid racemization. Now, this requires some biochemistry. Let me just briefly go through it. Amino acids you've probably seen in biochemistry somewhere, you've probably seen the representation of amino acids. And the funny thing about amino acids is that they can exist in two mirror forms, right? Don't push me too much on biochemistry, but the idea is that those complex structures of the carbon and then the associated radicals and the amino radical and the acid radical, this configuration can exist in two mirror shapes, right? One is called the L shape and the other is called the D shape. Now, the funny thing is, living creatures only use the L-shaped amino acids for some reason, and they're very peculiar with this. So, these amino acids tend to decompose or to change into their D shapes, and for some reason these are eliminated by the organisms, by the living beings. So this means that inside our bodies, all our proteins are made of L-shaped amino acids. Now, when animals die and there remains the proteins that are not completely destroyed are then gradually changed, their amino acids gradually change to the D shape. And after a certain period, there will be a balance between L shape and D shapes. Okay. So this is called racemization, amino acid racemization. And this technique has been used to date right whales, bowhead whales, from their eyes, from the protein in their eyes. Again, the animals are dead, are killed, and the studies have been done with bowheads captured in Greenland by Inuits, by Eskimos. And so they take the eye, and because the eye has layers, because the eye also grows, right? As the animal grows, the eye grows, but the eye grows by adding, in this case, the lens of the eye is made of protein, and it grows by accretion, so you have rings. And what they do is they go to the inner eye lens, inner circle, and extract proteins there and measure the racemization of aspartic acid, which is one particular amino acid that racemizes very quickly. So basically from this, they have been able to determine that bowhead whales can live up to 100 years. And so it's possible that the age estimates that we have for other whales are also undervalued, and that whales can live certainly up to 100 years and maybe more. So that's one element. And then this was the graph I was talking about. This is the graph that shows how animals, the relationship between age and size. Okay? If we know the age of animals, even if they are dead, then we can correlate the age that they have with the size that they have. and we can build these kinds of curves. So these are you have the curves there for northern elephant seals, and you have the curves there for sperm whales, and you see that they are very different from what you would expect from a mammal, right? Certainly very different from our growth curve, where you have a very rapid growth when you're young, and then you stabilize when you're in your middle age, and as you move toward the old age, like I am, you can see that your size is decreasing. Your size decreases. So that's very unusual for mammals, and it's probably explained, at least I explain this, based on the fact that they live in waters, so they're not constrained in their growth by gravity, like fish. So that's one element here. The other element here is also that, of course, there's dimorphism there, right? So you see males grow bigger than females, and we know in these two species, we know what this happens. And the other element that is important from these curves is that sexual maturity, because they grow continuously, sexual maturity is not connected with adulthood or the maximum size, like in many species. So many species, they are juveniles, and then when they reach their adulthood or they reach sexual maturity, they stop growing. This does not happen with many marine mammals, in particular these cetaceans. The final element that we can take from this graph is delayed sexual maturity, right? You see there, and this is a tricky question for you, Mark. You see there that female sperm whales and female elephant seals mature before the males, right? So, and the question for Mark is, this is not common to all the marine mammals. It happens in marine mammals where you also have this dimorphism, where you have big males and smaller females. And it means that in some way, males in these dimorphic species delay their sexual maturity. Mark, can you guess at why this should be so?
Maybe because they fight each other. So if they grow more, they have more chances to win.
Yes, good, very good, absolutely. What you have here is so this is genetic, right? So sperm whales mature when they're about 15 or between 15 and 20 years, whereas females mature before they're 10, right? So this is genetic. It's not something that it's not that the sperm whale says, an individual sperm whale says, well, I'm too small, I cannot fight the big males, so well, I'll live without sex for a few years. Now, this is just something that it just doesn't happen. The animals live while their sisters are already reproducing. The animals have not reached their sexual maturity for 10 years, right? So this is genetic. But the basis is what you've just described. It's a selection pressure. So animals that mature sooner have very little possibility of competing with the big males. Whereas animals that mature later, they can then focus their energy on growing, and they'll have more chances of reproducing. So the result is a selective pressure to delay sexual maturity and focus the energy on growing. Okay. So, but this other line here, and this is probably, this is almost the last slide. These things so they have a genetic element, but they also they're flexible, right? And for instance, we see in this graph an indication that the mean age of sexual maturity for female whales minke whales, fin whales, and sei whales, decreased last century in correlation with hunting, right? So this also shows that there is, so the obvious reason is that there being less whales, they responded by having an earlier sexual maturity. So this means that maybe there has to be some kind of social control of sexual maturity, in the sense that if we're talking about females here, in the sense that if females are with their mothers or their aunts, or they have other females around, they, for some physiological reason, tend to delay their sexual maturity. Whereas if they're alone, and if the population density is smaller, these social controls are relaxed, and the whales mature earlier. So you have this interesting play of genetics and social control. And the last slide, the really last slide I have here, is this one on death, right? So even if whales are not hunted, even if seals are not culled, these animals die, like all of them, and they die from there are many causes of natural mortality. Okay? They do have diseases, and we know they have diseases. They have bacterial diseases, they have viral diseases. They can be affected by toxins, even natural toxins. Say, for instance, for red tides, so the big accumulation of dinoflagellates, so phytoplankton, that is toxic, and these high concentrations of these toxins will get into the water, will get into the food chain, and whales are, well, marine animals in general, are known to be affected and to die from these red tides. And occasionally, even in the Azores, we get dead animals that are likely that have died from an infection, right? And that's number one. And that's, let me open a parenthesis as an advice. You should be very careful when dealing with stranded animals. If an animal died and if a dolphin or a seal is dead on the beach, it may mean it died from natural causes. It may mean that it has infection by bacteria, that it has infections by virus, and as we all know, bacteria and virus jump from animals to humans. So if you see a stranded animal, don't touch it and if you have to touch it, use gloves, use the precautions, use a mask, because they may contain dangerous pathogens. Of course, animals die from interspecific competition, although I cannot give you an example of this. We know that the mortality may increase theoretically in very dense populations, but I don't know of any demonstration of this. And of course, there are predators, right? And I would say that most causes of natural mortality would be diseases on one hand for old animals, and predation on the other hand, mostly for young animals, right? So you have, so I would say that most adult marine mammals are relatively immune from predation, but young ones are very sensitive to it, right? And they are targeted by sharks, by big ocean predators, being it sharks or being it other marine mammals. The main ones, Mark. what would you say is the main predator of marine mammals, which is also a marine mammal?
The Orcinus orca, the killer whale.
Yes, obviously. Do you know why it's called a killer whale?
It's called because the Spanish people call them asesina de ballenas.
Oh, you
But
know
they
that.
yeah,
You got me there. Good. Yes, please explain. Yes.
it's from asesina de ballenas, but they translate it wrongly. So instead of uh, killer
Whale
whale,
killer.
no, whale killer, they translate it to killer whale.
Yes. So that's a good point. I mean, the idea is that we have now is that the main target of orcas were whales, were whales with other cetaceans, and so and we have records from old travelers and even old whale hunting logs where they would see and they would record packs of orcas attacking other cetaceans and even whales. They would be targeting mostly the juveniles, but that was their main focus. So they were actually whale killers, and so the big industrial whaling also disrupted the food chains from orcas and is responsible for other, we could probably discuss other implications of this later. What I wanted to say now, just to going back to the orcas, is that it's probably orcas that were, or their relatives, that were responsible for the current pattern that we see in many baleen whales of moving up and down and doing these huge migrations from the Arctic to the tropics and back again, right? And the idea that there's a theory, there's a paper that puts forward this idea that whales move to the tropics to give birth, to stay away from the predation of orcas, so that they can have their calves, and that they can feed their calves until they're larger and less susceptible to predation. So it's a funny, it's interesting to think that these huge migration routes of baleen whales that are so characteristic of baleen whales evolved as a response to predation by orcas. And that's the last slide. That was the last point I wanted to make about all these issues that have to do with the population dynamics of marine mammals. I'm going to stop this. I'm going to stop the recording.